Chapter 6, Introduction. Name of this chapter is A Tour of the Cell. We'll start with few discoveries. Robert Hooke, as you can see, in 1600s, centuries ago, he made first microscope with stone and carved glass pieces and some candlelight. And then came these discoveries. He looked at the cork bark of the tree and he said that it looks like there are compartments in them. At that time, we didn't know what cells are. That is where the journey started. Curiosity of a human being takes us to the journey of miles and miles and eons sometimes, right? And still we don't know much about a cell, by the way. Claydon and Schwann, they said that, hey, all living organisms are composed of cells. That means viruses, are they not living organisms? Scientists are still debating that, by the way. We don't know. Some consider viruses as organisms and some say that they're not. They don't even have cells. Virtue, what did he say? You guys can read. As you can see, everything is made of cells. Animals, plants, mushrooms, fungi. The bread mold that you see, yes, that is also made of cells. That's the importance of cells. If you're looking at a living organism, then it must be made of cell. What is a cell, right? So a cell is an organism's basic unit of structure and function. Make sure that you will be able to say it, okay? See, a person is a unit. We have wholesome thinking, working, feeling, everything, digestion, everything in one structure, one unit. That is why it is human. Like that, we humans are made of cells, which is a unit, which is a working, functional structure, which has everything in it. It thinks, it works, it is born, it dies, everything. Make sure you memorize these two statements of cell theory. All cells come from pre-existing cells. It's the next statement. This number varies, guys. Human body is made of 50 to 100 trillion cells. And there are 200 different types of cells. Nerve cells, which make the brain. Muscle cells, which make our strength, right? Different sizes of the cell. Yes, the smallest cell could be 1 micrometer, it seems, which is bacteria. Yes, bacteria are cells. Different types of cells. These are plant cells, bean-shaped, 2 stomata cells. This is what my research was in. Paramecium, algae in water. What do we use microscopy for? Microscopes are those tools which are used by scientists as a ruler. We use a ruler for measuring the height of a person, length of a person, width of a person, right? All these like that. We use microscope to measure the length and width of the structures which are very tiny, which we can't see with our naked eye. Light microscope uses light, electron microscopes use electrons, some other microscopes use other sources in order to make an image, like laser microscope, UV microscope, yes. But then if UV is used to look at the structure on the slide under the microscope or under the magnifying lens, then you will not see it directly, you will see it on the computer, okay? In lab, we memorize all the structures of a microscope and then we use and we also learn how to use them, which is totally an experiential session. That's why we need in-person labs. We look at eyepieces. into which the light is coming, and then there is a nose ring, the parts of the microscope in which the carved lenses are kept, through which the light diffracts and becomes bigger to make a bigger image in our eye. These are called objectives. Under the objectives, there is stage on which we place the specimen. Under that, there is condenser, then field diaphragm, and then a mirror on which the light actually comes from the bulb, which is in the base of the microscope. Make sure you know all this path of the light of the microscope. From eyepiece to bulb, bulb to eyepiece. So both ways. What are the two properties of a microscope? Magnification means enlarging the image, not the specimen itself. Resolution, clarity. If there are two dots, then they should be seen as two separate dots. If it looks like a blotch, then it is not a good microscope. That is called resolution. Contrast is important because many structures under the microscope, they are colorless. Many organisms have colorless structures. In order to see them, you need to create contrast. And that is done by most of the time using the dyes, colors. Okay. Electron microscopes are of two types. Transmission electron microscope, they show you internal structure of the cells, and scanning electron microscope shows a 3D image. As you can see, this is the cilia, which are the bristles in our trachea, hair-like structures which beat upwards. They are beating all the time. They're beating, okay, they're moving. Each one is made of membrane and the protein fibers inside. That is what you can see using the TEM. SEM for 3D, TEM is for internal images. How can we separate the structures of a cell so that we can learn in depth? That is done by using a centrifuge. You might get a chance to look at these in the lab. Cells are of two types, prokaryotic cells and eukaryotic cells. Karyon means nucleus. They were formed without a proper packaging of the DNA, which is material which gives them inheritance characters. Bacteria and archaea, they come under prokaryotic cells. That means simple cells. They don't have many compartments inside. Eukaryotic cells are further categorized into protists, fungi, animals, and plantae. Eu means true. Karyon means nucleus. There are structures which are made of plasma membrane, have a fluid inside called cytoplasm or cytosol, and DNA is there, and there are some ribosomes. These are the basic structures found in a cell. These are there both in prokaryotes and eukaryotes. If there was ever, or if there will be ever a question which asks what are the basic structures in a cell, make sure that you name all these four. And prokaryotic cells, they do not have nucleus, but then they have compressed nuclear structure, not nuclear, nucleic acids material. It is called nucleoid. They don't have nucleus. By the way, this is bacteria. Bacteria are cells. They don't have organelles according to some textbooks, and according to some other textbooks, they do. What are organelles? Organelles are those structures which are made of membrane, contain space inside, or maybe fluid, and or fluid also, and reactions take place inside. Then it is called an organelle. Bacteria do not have organelles according to us as well, according to my tests. Ribosomes are not organelles. They have more structures than these. Eukaryotic cells, they do have more than nucleus, mitochondria, and chloroplast. Other organelles in eukaryotes are endoplasmic reticulum, which is of two types, rough, which has ribosomes studded on the outside, and Golgi, and lysosomes. And what else? This is smooth endoplasmic reticulum. This is rough endoplasmic reticulum. This is Golgi apparatus, and lysosomes, peroxisomes. These are all the organelles. Again, nucleolus is not an organelle. Flagellum is not an organelle. Mitochondria, which are kept in place by microtubules, they are not organelles. Why is the surface area to volume ratio is important? It should be higher if it was bigger size cell, one cell making up the membrane, then this surface area won't be enough for the entire volume of the cell to provide the nutrition, oxygen, and all the glucose and amino acids and vitamins and minerals that it needs. So that's why if the brain is made of multiple cells, then their surface area is enough for each volume. That is why surface area to volume ratio must be higher. Memorize that, understand that. These are the logistics of the cell. Let's go into the cell organelles, each one now.